Good afternoon, everybody. It's my pleasure to introduce the first of our two speakers today. Peyton Palsha is going to start us off today. Peyton is a native of Florida, but she got her undergraduate degree at the University of Arkansas. Big Suey, everyone. Peyton, in addition to getting a degree in horticulture, was on the swim team there. Then she came to the University of Georgia a year ago, was on the swim team for 1 year, trying to balance athletics and academics, and now she is a full-time graduate student. She started out, and she'll talk about this a little bit, last year doing a joint project with Dr. McGregor and myself, and now she is working on hydroponic lettuce production, looking at the effects of light intensity and potassium nutrition. And with that, Peyton, it's all yours.
Hi, I'm Peyton. So today I'm going to be talking to you about some of the research that I've done so far. So I've been here for about over a year. But overall, the end goal is looking at hydroponic lettuce in response to potassium and photosynthetic photon flux density. So for introduction, I'm going to start with controlled environmental agriculture. It's important to note that electricity costs to provide lighting in controlled environmental agriculture is quite high. The efficient use of light is critical for profitable crop production, and crop growth depends on the amount of light captured by the canopy and the light use efficiency. As you can see from the bottom, electricity costs for supplemental lighting in controlled environmental agriculture cost about $180,000 a hectare a year, which costs growers about 25% of their annual revenue. We are using a hydroponic system for my experiment. Hydroponic systems are soilless growing systems that stimulate plant growth conditions by using a water solution and nutrients. Growing lettuce hydroponically allows better management of nutrients pH, increase in water use efficiency, higher production turnover, as well as a reduction in labor costs for weeding and soil preparation since there are no weeds for soil. On the bottom, I'm using is an example of what I will be using in the greenhouse. Eventually, we will use a styrofoam platform and net for the netting for pots. The plants to go through, they sit in a nutrient solution with a bubble stone and an air pump to give them air. I'm using a multispectral imaging system to calculate a lot of my data. This imaging system is a fast and non-invasive way to quantify physiological parameters like canopy size and even identify some stress symptoms. The multispectral imaging system has about 8 channels that generate different monochrome images from different colors of LED light. There's 1, 2, 3, 4, 5, 6, 7, 8. And then we also get a chlorophyll fluorescence image. The chlorophyll fluorescent image creates a masked image. Okay. This masked image helps create the RGB image, which then when the background is removed, all that is left is the plant. This chlorophyll fluorescence can be used to quantify canopy size or detect stress. We run images through a program called Python, which will analyze images to import the results for us. But that's not mine, that's Mark's, and you'll hear him talk about more of that next. So for an overview, the first thing I'm going to do is I'm going to talk in chronological order of what I have done so far and what I am currently doing and what I plan to do moving forward based on chapters. For the first chapter, we would like to investigate the potential use of a multispectral imaging system as a screening tool for early detection of zucchini, yellow mosaic virus, and squash, which have been completed, and I worked in joint with Dr. McGregor and Dr. Van Ursel. The next one, evaluate the growth, morphology, and physiology of hydroponic lettuce in response to photosynthetic photon flux density under sole source lighting. That has been completed. And we are currently in the process of working on almost the very same experiment, except this time we're looking at hydroponic lettuce in response to potassium under sole source lighting. Eventually, we'd like to move on to investigate the growth, morphology, and physiology of hydroponic lettuce in response to both photosynthetic photon flux density and potassium in a greenhouse environment. Chapter 1. So for the multispectral imaging as a potential screening tool for early detection in squash, we will evaluate the potential use of this multispectral imaging system as a screening tool for early detection of zucchini mosaic virus in squash. The materials that we use Dixie yellow crewneck squash grown in a growth chamber under a 16-hour photo period. The treatments we used, they were grown at a ppf of 350 micromoles per meter squared per second under sole source white light. We had 15 plants that were inoculated with the ZMV virus from infected plant tissue that was inoculated. And then we had 15 plants that were not inoculated, and that was our control. Measurements that we took were chlorophyll fluorescent intensity and normalized difference vegetation index. Normalized difference vegetational index and DVI for short, as well as chlorophyll fluorescent intensity, CFI. Those are the calculations that we got for our data. So for the chlorophyll fluorescent intensity, we saw we used so we have the control from day 8 and the control from day 9, from day 8 as well. We were able to see a peak in chlorophyll fluorescent intensity, and I believe you can see that there are 2, no, that is not the right one. So, but anyways, we're able to, I'm not quite sure on that completely at the moment. And then normalized or NDVI index, we saw 2 peaks. We were able to see that because the first peak, this first peak is basically the greenness in the plant, and then this is basically the yellow, and there seems to be a 3rd peak down here. It might be like the cotyledons, but you can see that there are different intensities of greenness within the plant. This NDVI showed us that because there were different colors from the plants, which may have been a result of a nutrition deficiency earlier on in the study, we got we got a different the standard deviation. It was important between the control and the inoculated because there was such a difference within color of the chlorophyll fluorescence. But overall, the conclusion of this at the moment is that the use of this multispectral imaging system as a screening tool for ZYMV is yet to really be determined. Chapter 2, the effect of PPFD on lettuce under sole source lighting. The objectives that we looked at, we want to evaluate the growth, morphology, and physiology of hydroponic lettuce. The response to PPFD under sole source lighting. We investigated how the different PPFDs affected lettuce growth, morphology, light capture, and the light use efficiency. The materials we used cultivar Rex lettuce grown in a growth chamber under a 16-hour photo period. We grew it under different PPFDs ranging from 201 to 413 micromoles per meter squared per second. And it was all under sole source white LED lights. The measurements we took were projected canopy size throughout the growing cycle, dry and fresh root and shoot weight. The calculations we used to get our data: total incident light, specific leaf area, and light use efficiency. So as you can see, we grew some of the seedlings in the walk-in growth chamber, and then when they were ready, we transplanted them into their individual growing systems. They're in individual buckets because the airflow in the growth chamber comes from the bottom, and if we use a full, covered up all of it, there would be no airflow. As you can see the maybe after like a week of growing, you can see some of the seedlings start to grow, and then about halfway, we did weed out some of the lettuce because they obviously got too big and they were so close. We decided to weed some of them out, at least half every other, so they would not be competing against each other for light. So some of the results, we got root and shoot weight versus PPFD. As you can see, the root and shoot dry weight increased under a higher PPFD, and then the fresh weight responded quite similar. We have the shoot dry weight versus total incident light. We have for high PPFD, increases incident light, but that's not shown. Total incident light strongly correlates with shoot dry weight, and the light use efficiency has not actually been affected by PPFD. The conclusions were that under lower PPFD, plants increased specific leaf area and projected canopy size to increase light capture, and light use efficiency was not an important factor in determining growth. But growth and total incident light are strongly and positively correlated. For the second chapter, we are going to look at the effect of potassium on lettuce under sole source lighting, and that is currently in the process right now. We want to evaluate the growth, morphology, and physiology of hydroponic lettuce in response to potassium under sole source lighting. So we will investigate how different rates of potassium will affect hydroponic lettuce growth, morphology, light capture, and light use efficiency. So what is potassium? Potassium basically is known for promoting cell expansion and leaf elongation within plants. So if potassium can increase leaf elongation, then plants should be able to increase the amount of light captured by their canopy, right? So since crop growth depends on the amount of light captured by the canopy and the light use efficiency, an increase in leaf elongation should positively affect overall crop growth. And compared to lighting costs, potassium is much cheaper. We use, again, cultivar Rex lettuce grown also in the growth chamber under 16-hour photo period. The treatments we did was grew lettuce under different potassium rates, potassium rates using potassium sulfate. We were able to get 100 to 250 milligrams per liter of potassium in each treatment. We also grew the lettuce under PPFD of 250, and within that variance between under the plants in the growth chamber, we got 191 to 247 micromoles per meter squared second. It was also under white LED light. Again, the measurements and the calculations are the same for this experiment as well. So again, transplanted them into the hydroponic system. As you can see in the middle picture, the light is all the same. Originally, in the previous experiment, when we looked at PPFD, we had to turn some light panels off. Well, they're all on this time. And they're currently growing. We did have a little bit of a mix-up, so we're gonna try it again on Friday. The effect of PPFDs, the overall goal is to look at the effect of PPFD and potassium on lettuce in the greenhouse environment. So the objectives moving forward, what I would like to accomplish, we want to evaluate the growth, morphology, and physiology of hydroponic lettuce in response to PPFD and potassium in the greenhouse environment. So by investigating how different PPFDs and rates of potassium affect hydroponic lettuce growth, morphology, light capture, and light use efficiency. Material methods, again, will be the same. Rex lettuce grown in growth chamber under a 16-hour photo period. The treatments, we will grow lettuce under different potassium rates, under different PPFDs, and this time they're going to be grown in the greenhouse instead of the growth chamber. We will also be doing the same measurements and calculations for this experiment as well. So this is exactly what I wanted to show you. Instead of the little greenhouse we saw, I had 32 plants. We're going to have way more, obviously. So this is from one of my colleagues, TC. He looked at something similar except nitrogen. And this was his study in the greenhouse. This is what my project will look like, quite similar, but obviously with different treatments. I just wanted you guys to understand maybe the difference in like the way to look from the growth chamber and into the greenhouse. So the end goal of this is to identify optimum PPFD target and potassium rates of hydroponic lettuce for maximum production efficiency. Determine if the supplemental potassium will increase light captured by the plant canopy and increase the light use efficiency without the additional increase of supplemental lighting, thus lowering electricity costs. That's it.
Do you have any questions, comments, suggestions for Peyton?
You know I have a question. Is this affecting the virus spreading?
Of course. Yeah,
I just want to make sure I understand.
yeah, of course.
Right, so that maybe the name, the other matrix.
Is it this
No,
one?
no, the other,
Oh.
yeah.
This one? Okay.
Yeah, I think we kind of talked about this over email, but I'm not quite sure I understand it correctly. So the standard deviation, you have your control is higher than your inoculated, and so that standard deviation is basically a measurement, if I understand correctly, of variation in pixels. So the more different the pixels are, the higher the standard deviation would be. So I guess I'm curious why the healthy ones have more variation than the.
So I did go back and look at a few. What it seemed, I'm not sure if it's for NDVI specifically, if it was DFI, but it almost captured also the cotyledons, and they were yellow. And as they started to like die off, it's still like it almost like it decreased. It went up, and then once it got to the peak, it seemed that it was picking up the cotyledons. And therefore, when the cotyledons were like almost dying off, it seemed as if that's why it thought that maybe there were less pixels specifically. I'm not necessarily sure. We're still gonna have to go back and look at like the legit like images, but from what it looked like is that it was picking up the cotyledons, and as the cotyledons were dying, it also thought that there were less pixels.
But the cotyledons are also dying in the controls, right?
Yeah.
But it looks like they may have some that's later in the control plans and then the inoculated plans. We don't know that for sure. We need to reorganize the pictures to be able to look at that. It's certainly very counterintuitive. Do you want
So
to
can you
describe
go
what
to
the
the previous
sentence?
slide? Like,
Well-
that they talk about senescence, go back to the previous slide. Because there is a decrease in chlorophyll intensity, does that mean that they are senescing at that point, or like on day, like the curve looks like it's going up and down, right? One
Yeah.
thing is,
But see, in this one, the controls are actually lower, which is what I would expect, I guess. In this, for the standard deviation, right, the control standard deviation is lower. And that's what I would expect, less variation in the control than in the.
Peyton, I think it's important that you describe the symptoms of this
Okay.
virus, because otherwise none of this will make any sense to anyone who isn't familiar with the virus.
Okay, so some of the symptoms of the virus are fluorosis, I believe. And one of the issues we had was either nutrition deficiency or humidity issue. But I did not immediately water with fertilizer water, and then I did start watering with the fertilizer water. So we weren't sure if the fluorosis we were seeing was an issue with the actual virus, or if it was just like a nutrient deficiency issue.
I think the key thing here is the virus's name is zucchini yellow mosaic virus. So it makes like these kind of splotchy mosaic things on the leaf. So it's not uniform chlorosis.
Yeah.
Green and yellow splotches.
Yeah. So
I would think that would cause a lot of variation.
yeah, we'll have to go back and look and see
I'm just
specifically why.
curious.
Yeah, you're okay. I definitely think it's worth more looking into. Just really have not gotten a whole lot of time for it. Any other questions?
I'm also wondering if, whether you can separate out, like, if it is like, just the cotyledons that is interfering, whether when you get these images, you can subtract that out.
Can we subtract that out currently?
Certainly not currently. That would not be easy to do, I don't think. But so, in these two images, you can see the cotyledons, and in the inoculated plant, the cotyledons are almost black. They have practically no chlorophyll left. And I mean, it's little, if you don't know exactly what you're looking at. In the control plant, you can see part of the cotyledons pretty well. Yeah, but even there, and some of it apparently no longer has any chlorophyll and is not fluorescing anymore.
Yeah, so the other thing to remember that it's the cotyledons is what we are
Or
inoculating on it,
Yeah.
right? So we're actually damaging the cotyledons.
Yeah.
Because we're inoculating on it, so, and we do the cotyledons so that we don't damage the true leaves,
Okay.
because that will cause even more problems.
But
I mean,
the
still-
opposite thing, you need the.
Yeah. But still, the damage on the inoculated and non-inoculated should be the same, because we do it exactly the same.
Except for the virus.
Except for the virus, but still the grid, still a plant, it's not the non-inoculated, it's mock inoculated. So except for that the plant that we're inoculating with is not infected with the virus, everything is the same.
And we certainly saw the typical symptoms of the
Yeah.
virus in these plants.
Yeah.
We have color images of
Yeah.
all of these plants for each day.
Mhm.
I'm just curious because, Jorge is doing something with the imaging also. So
Yeah.
just want to make sure I understand what is happening.
Any other questions? Comments?
Yeah, maybe I missed this in the beginning, but how did you decide to look at I mean,
Because Caleb started with a K.
Dr. Mark, honestly.
No, but- I thought you gave a pretty clear reason.
Yeah, I mean, the reasoning is obvious, but I don't know if I would have gotten to it
Well, you may
immediately.
want to repeat it because maybe you should have catch it.
Oh, okay, so, potassium, we are doing potassium specifically because it increases leaf elongation and cell expansion, and our hope is that if the leaf elongation with potassium, more potassium, and the leaves become more elongated, they may be able to capture more light under maybe less light levels. And that is maybe, hopefully, we can decrease the electricity cost and like growing like production. But we're just gonna look at, I guess, what specific rate of potassium as well as the specific PPFD it works best under to get the best crop growth.
Do we have any questions from anyone not in the room here, anyone on Zoom? If not, Peyton?
Yeah, I had another question.
If not, Peyton, you're not off the hook yet.
Go ahead, I still have 6 minutes.
So for the potassium study, how often are you going to change the solution, like the hydroponic solution?
We're not sure yet, but I know that in the last study, when we just looked at PPFD, I did have to like top off some of this like nutrient solution like base, but then we hadn't included potassium, like extra potassium into it. But I know that we obviously had to adjust pH, but we'll have to talk about that because I'm not exactly sure how we would go about like fixing it as we go forward. I don't know if we would add any more. We would just have to fix like maybe the base or the pH.
Yeah, so we don't replace the solution ever. We just add more to it.
Okay. So,
Yeah.
And then we do pH adjustments because at some point pH is likely to go up really quickly.
That happened in our first study of looking at PPFD. I had to redo it. I had to read it twice. But first time we had a bunch of micronutrient deficiencies. The pH was quite awful, quite high. And we had a lot of tip burn and like issues and fluorosis in the actual plants. I didn't show you that. I showed you the nice version. That was we didn't realize we needed to adjust pH on us.
I don't know if you're planning to just do some nutrient analysis for the leaves or even measure, just to keep track of the hydroponics, just measure the solution itself. I know that there is at the College Station, right, we have a facility that does nutrient analysis.
We did a nutrient analysis on the plant leaves, like after we harvested them, I believe. But I don't think we've done it for the nutrient solution that's left over at the end of the study now.
And if you go to the nutrient analysis, don't use the UGA lab. Thanks, they're not twice as expensive as the commercial lab. It's unbelievable. And was slow too. Yeah. I don't know how they managed to do this.
So do you send it to the water lab?
Yeah. Yeah, much bigger.
Hey, I had a question. We're over here in Griffin. Can you hear me?
Yeah, I can hear you.
Okay, this is Hannah Cowart from the Griffin campus. I had a question about your methodology. I think it's in chapter 1 about using I think you called it like growth chambers, basically the buckets instead of just the styrofoam sheets like your friend did in his experiment. But you were saying something about airflow being greater in this case, and I was a little confused about what you meant by that.
So what I meant by that is that instead of using like, we used the buckets because in these growth chambers, the airflow came from the bottom of the grate. So in order for us to be able to have that continued airflow within the growth chamber, we had to put them in buckets versus like one big tub. As you can see in the greenhouse that my colleague did, they're bigger. They're like these big rectangular or like square tubs, and it's like one nutrient solution. All of the specific amount of the lettuce is in just that treatment of nutrient solution. So obviously you couldn't put like one big tub and all of the lettuce within the same exact nutrient solution because we would cover the bottom of the growth chamber, which we wouldn't be able to have as much airflow through the bottom, and that's no bueno.
So now when the convirant guy came and the media says, oh, we should never put something directly on that, you have to have a rack above it, like, like.
Yeah. Like a little step stool type of.
Well, it's actually racks that put in those. We actually have one of our growth chambers have them, the other ones have all disappeared.
Oh.
So it's like a refrigerator rack,
Oh, yeah.
and we shouldn't be using the bottom as a
Yeah.
shelf, I guess.
That's funny because every confinement growth channel I have ever seen them use as plants, like on the bottom.
So that's what we did, but I said, when the guy came and retrofitted this one, and actually, so there's a rack we put above it.
No.
But so we do try to make sure that we have enough space for adequate airflow.
Yes, and then when I will do the potassium study, if I can find it, I took out the picture, but we used 2 growth chambers this time basically, and we spread out the 32 plants, so we don't have to weed them out halfway through, so there's probably more airflow.
And with that, Peyton, I am afraid you are out of time.
It's Mark's turn.
Thank you.